The overall goal of this protocol is to demonstrate how to dissect the Drosophila eye antennal imaginal discs and brains carrying genetically defined fluorescently marked clones, and how to process them to visualize and quantify gene expression changes and cell invasiveness.
The described procedures can help to provide novel insights into the role of genes in epithelial development, homeostasis, or disease, and to dissect mechanisms underlying cell competition, compensatory proliferation, or interclonal cooperation. The main advantage of the study is that we provide an entire suite of protocols tailored for in-depth quantitative and qualitative analysis of the Drosophila genetic mosaics.
Generally, individuals new working with the Drosophila model struggle because they cannot recognize the eye antenna imaginal disc from the other imaginal disc within the Drosophila larva. To ease the recognition of eye antenna imaginal disc bearing fluorescently marked clones within the larval body, it is recommended to perform the initial dissections under a fluorescent microscope.
Mosaic analysis with a repressible cell marker, or MARCAM, enables the generation of genetically defined clonal patches in an otherwise phenotypically wild-type context. A flip driven by an eyeless promoter catalyzes recombination between two FRT elements flanking a stop cassette marked with a yellow gene. After DNA replication, flip catalyzes the exchange of the non-sister chromatids between the homologous chromosomes, generating sister chromatids, one carrying the wild-type allele and GAL80 repressor, and the other the mutant. In mitosis, these segregate to produce two daughter cells, one being a homozygous mutant and the other wild-type.